Hello, my name is Augusta Udo. I am from Nigeria, and here with me is Mr. Daniel from Kenya. Together, we are going to be presenting on the topic, should African governments invest in genetic research for national security purposes? Example, bioterrorism defense. We are from the Affirmative Team. Introduction. In this contemporary world, the scope of national security threats extends far beyond explosives and firearms. There is, however, a new front of warfare that is covert, less certain, and exponentially more dangerous, which is the biological warfare. For proactive measures, African authorities should prioritize genetic research as an urgent matter of state security. These are our opening statements. Today, we strongly support the motion that African governments should invest in genetic research for national security purposes, example for defending against bioterrorism. Okay, these are our constructive arguments. Now, genetic research enhances by national biosurveillance and early threat detections. We can see this in the Journal of Medical Virology that explained that genomic surveillance uses sequencing to track pathogen evolutions and monitor outbreak. This was first applied in Ebola and it was later used to detect COVID-19 variants in Nigeria and South Africa. And we have the real-time genomic sequencing to improve outbreak responses. How is this happening? Real-time genomic sequencing allows scientists to analyze pathogens' complete DNA or RNA as an outbreak unfolds, not after. Please underline that statement, not after. This helps identify who is infected, how is it spreading, and whether it is mutating, while there's still time to act. We can see this in COVID-19 vaccine. We can take it as an example that they were created within months after scientists sequenced the virus. And Kramer explained it in Nature that this was very possible, and this was done because they had the virus full genetic code early on. So with real-time sequencing, the scientists were able to have the full genetic code, the virus full genetic code early on, so as to produce or develop vaccine for it. We have genomic and trace the source of bioterror threats. Yes. Now let's look at the attack on USA, the 2001 anthrax attack in USA. They received a letter containing this anthrax spore. That was in October 2001. They received a letter containing this anthrax spore, and they sent it to them through their USA portal system. People that received the letter were the news media officers and two senators, Senator Tom and Senator Patrick. They received this letter and they opened it. So when they opened it, they found out that the letter contained a powder with anthrax spore. And inside that letter, there was a threatening message like, "You can't stop us. We have this anthrax." So that shows that it was a proper terror attack on the US people. And they contacted the disease, the anthrax disease, because this anthrax disease now is anthrax is a deadly bacterial. It can be contacted through inhalation, that is breathing in, through skin to skin contact, could be through contaminated food. So in this case, they contacted it through breathing in, and then they spread it. Along the line, 5 people died, 17 people got sick. So there was the country was destabilized. So what happened now is that they started pointing accusing fingers to other countries that they could be the one to launch this bioterror attack on them. But then genetic research came in to help. What they did was that scientists used genomic sequencing to identify the exact strength of the anthrax used, and they now trace it back to the origin. Like I said, genomic can trace the source of bioterror threat. So they trace it back to the origin of that attack and discovered that it came from the US government laboratory. And this information, these genetic informations now gave the FBI glue on how to carry out their investigation, and they still narrowed down the investigation to the lab, and they discovered that one Dr. Bruce Ivins has already worked on that same strand of anthrax, and he became the main suspect. Why this matters? It matters because it showed how bioterrorism can be used to spread fear, can be used to disrupt country economy, can be used to destroy lives, and then it boosted the use of genetic tools to track and fight biological threats. So in Africa now, if we have the genetic tools, if our genetic research lab is equipped with the necessary genetic tools, Africa can track and fight any bioterror attack that comes into the continent. That rapid diagnosis using CRISPR and sequencing tools. It means using advanced tools like CRISPR. No, CRISPR is genetically modified. It's a genetic tools. It's a genetic tools. So using advanced tools like CRISPR and DNA sequencing to quickly find out what disease is making someone to be sick. Yes, and it has been used to identify diseases like the Ebola, the COVID-19 before now, and from this makes them perfect for field use during a bioterror event where traditional labs may not be available. And also, bioterrorists now, they can decide to use genetically engineered bacteria or viruses. So with the help of CRISPR, it decides to be able to identify whether the bacteria or the viruses is genetically modified or not. Then vaccine design through genomics, that is reverse vaccinology. Now, before designing a vaccine, scientists need the access to the full genetic code of that particular virus that they want to design a vaccine for. Here in COVID-19 vaccine, we can see that it was Moderna were developed in record time within months, because scientists had access to the full genetic code of SARS-CoV-2 virus early on. And Grammar explained it in Nature that this was only possible through genomic sequencing. If there was no genomic sequencing, they would not be able to have the full genetic code of SARS-CoV-2 virus. And if they did not have the full genetic code of SARS-CoV-2 virus, there will be no vaccine production. We have reduced reliance on foreign labs. Yes, when Africans are African governments equip their genetic research labs, there will be no need. This will improve independency and strengthen the national scientific capacity. We can see that during COVID-19, that the countries like Senegal and South Africa, they developed in countries diagnostic capabilities and genomic surveillance. And without that, they would have just had to be sending their samples outside country for analysis, for diagnosis, and this causes delay. And enable faster localized response. Now, when sending your sample outside the country for diagnostics, testing, and analysis, it is done on first in, first out. You met someone there, you have to wait for that person to finish his or her own analysis before your own. While there's outbreak in African country, we are queuing in international lab, waiting for first in, first out. Now imagine if in Africa hospitals and genetic research laboratories, we have equipment like the Illumina, the Oxford Nanopore. It will be very simple to analyze our sequence and carry out the outbreak swiftly without waiting for results from the outside laboratories. Now, protect genetic data from misuse. Yes, genetic data is information about your DNA or about your gene, or yes, gene of people, DNA of people, or animals, or plants, or microbes. And they are like fingerprints, very detailed and very unique. A situation whereby this genomic data mistakenly falls into the wrong hand, it can be misused in dangerous ways. For if it falls into the wrong hand, into the hand of terrorists, they can use it as design a bioweapon to affect people with a particular genetic trait, for example, sickle cell. And also, if they have access to these genetic data, bioterrorists now, they can alter a known pathogen, so they evade existing tests or treatment. That means that when they alter that genomic data, that pathogens, that particular pathogens, before developing a vaccine, that the pathogen genetic information is being incorporated into the vaccines. So when they have access to alter that pathogens, alter that known pathogens, the vaccine, the existing vaccine, will not be active anymore. So this is our argument. Now, negative arguments will say genetic research is too costly and African cannot afford it. Yes, genetic research is costly, but the cost of inaction is far higher. You can, okay, let's look at during COVID-19, for example, in Nigeria, you hardly see a group of 2 persons. It was banned, no group of 2 persons. There was no church, there was no mosque, there was no market. It corrupts, it disrupts the economic system, it collapsed the national health care. Lives were lost, millions were spent, the government spent so millions to serve the citizens. But if they had established, equipped the genetic research lab before now, before that time, there wouldn't have been disruption in the economy, because what, they would detect it early and then produce vaccine for it. And we saw it in Nature 2021 that African nations faced severe delays in vaccine access because they lacked local genomic and manufacturing capacity. You can see that. Negative arguments: scientifically unsustainable. The claim that genetic research is scientifically unsustainable is simple false. Genomic has already helped African countries track COVID-19 variants. Like I said before, the South Africans, the Nigerians, they were able to detect that COVID-19 variants early because the African CDC equipped their lab with genomic sequencer. So you can imagine if governments equip our labs with genomic sequencer, they will be able to track any disease that comes into the continents. Negative arguments: it is a long-term investment. Africa needs immediate health care. No, genetics is not only for long-term, it has immediate life-saving applications like real-time genomic sequencing. During that COVID-19, during that Ebola, they use real-time genomic sequencing to identify and contain Ebola outbreak, even the COVID-19 outbreak. So it is both for long-term and short-term. Negative arguments: genetic tools are too advanced for Africa's current infrastructure. This is not true. Africa already has world-class genomic centers in Nigeria, South Africa, Kenya, and Senegal. So they were the one that even led in the early detections of that COVID-19 variant, like I said before. So what we are saying here is that Africans have the best research scientists. They has a world-class genomic centers, and all they are looking for, or what they simply ask, is that the African government should invest in it. Strategically misplaced. Okay, yes, Africa faces a lot of challenges like the poverty, the disease, and the climate changes. But how can African country eradicate these challenges? How can they get to the end of these challenges? This is where genomic comes in, because it directly supports solutions to malnutrition through what? Bioengineered crops. We can use bioengineered crops in Africa to eradicate poverty. And in case of infectious disease, genomic can, through genomic surveillance and vaccine, like I mentioned before, through genomic surveillance, monitoring the symptoms of that deadly disease, monitor it, detect it early, and have the access to analyze, to sequence the full genetic of that particular virus, then produce or develop a vaccine for it. That will take care of the disease. And public health, by detecting outbreak fast, That is when we mention real-time genomic sequencing. By detecting the outbreak fast, Africa can stop any kinds of disease. Not just that we have disease, we Africans should invest money in disease. How do we take care of this disease when we don't know the roots of this disease? And it's only genetic research that can help us to detect the roots of this disease and then provide a lasting solution to it. In short, genomic research helps us fight today's problems and prepare us for tomorrow's. That's not misplaced, it is forward thinking. Closing statements. Africa is endowed with genetic diversity. Wisely used and conserved, this can contribute significantly to enhance peace and security in the continent. This is our selected references. Questions and answers. Thank you very much for listening.